All right, sorry for the delay. Let's see if this works now. Hardwired with the cable. All right, great. Glad that worked. Okay, so we're going to go over genetics today and kind of like central dogma, transcription, translation, lac operon, and then hopefully that'll take us to about an hour, and then I can answer any questions that you've got. So we'll dive right in. So you've probably seen this before. This is considered like the central dogma of molecular biology, or really of biology in general. But the idea that we're going to go from DNA to RNA to protein as kind of like our products, we obviously know that somewhere in here, we're also going to have this form of mRNA, messenger RNA. And we also know that we can go from DNA to RNA, and that's called transcription. So anytime you kind of see transcription, you're thinking about DNA to RNA, and then RNA to protein being this term called translation. Unsure where they got those terms from, but we know that that's what they're called. That's what everyone agrees to call them. But we also know that we talked about from the first lecture that I taught, going from protein all the way back to DNA, right? And that was called like a cDNA library, or we could use a DNA library to do that. Sorry, I don't know why it's giving that little interference noise. And we also know that we can go from RNA to DNA, right? And that would be reverse transcription. And so we've kind of updated this central dogma of biology to look a little bit more like this. So we know that every time a cell is going to get ready to divide, unless it's a gamete or like a sexual reproductive cell, it has to replicate its own DNA and then kind of like go through the split process of mitosis. So this is kind of like involved in mitosis, right? Or even meiosis in the first phase of meiosis, meiosis 1. We know that we can go forward from DNA to RNA, and that is transcription, but we could go from RNA back to DNA via reverse transcriptase or reverse transcription. We know that RNA obviously can multiply itself. So we have RNA replicase, which is kind of helping in there. And then as we talked about, translation to protein, and we know that we're going to kind of go via messenger RNA. In order to get this, we could be using tRNA. We could be using SN, small nuclear RNA, or even ribosomal RNA. So we know that we have kind of some variation within here. And we talked about also like using experiments to go from protein back to DNA, from the mRNA back to DNA. So we have, we're able to kind of use this as our foundation for understanding how we can get from genetic information to different products, but how we can also kind of almost like in a way like hijack this system by using biotechnology in order to like figure out more and learn more about kind of what's going on. So I think the biggest things to kind of think about when you're thinking about central dogma, and anytime you get a question that's related to this part of the topic, that you are thinking about kind of like, who are the big players? Like, what enzymes are working here? Am I in a prokaryotic cell? Am I in a eukaryotic cell? What's similar? What's different? And just being really familiar kind of with all the key players. I think that oftentimes it'll say like, okay, so I'm in transcription of XX gene in the, Drosophila. You kind of should already be thinking about, okay, who are the key players in transcription? I'm in a fly, so I'm in an animal cell. What's going to be different? What's similar? And just kind of being able to already think about those things, because you'll see that most of the questions are going to focus on that, on who those players are, or what would happen if you knocked out one of those players. So we're going to go through, each of those. How can you guys hear the I don't know, weird sound that it's making? Is it really bothersome? I'm not sure I have like a solution though, but let me know. Okay, it's kind of like I don't know, it's a weird sound. That's how I can describe it. So we're going to start with this DNA replication, right? This is just DNA forming new DNA. So we're thinking about mitosis and meiosis 1. So we've got what we consider our origin of replication. And that is basically where we're going to start like unzipping and kind of like looking inside to make the copies there. So we have a few key players here. Helicase is going to be an enzyme that's going to unzip the double-stranded DNA. Now you can kind of see this going in both directions. It's in green in this picture. And it's really kind of creating separation for all of the other enzymes and all of the other players to actually come in so that they can kind of like get in there and start reading the strand. We also are going to have some binding proteins that are just going to help stabilize it. And that's important because we know that on our kind of like facing sides here of our DNA strand, there are obviously the bases, and they usually are interacting with each other through like hydrogen bonding. And so they want to be close together. And so we actually need to add something. Whenever we add something, remember, we're thinking about a conformational change, and that's going to help keep them separated. So now that we've got them separated, we are going to then actually add in another enzyme, topoisomerase, or gyrase, as sometimes it might be referred to. And this is going to be working like on the outside. Basically, if you think about it, if you're unzipping kind of this already like helical structure, when you're pulling that apart, you're basically squashing everything at the ends. And so topoisomerase is going in and trying to like make sure that that's all smooth and it's not getting extra kinked. If you've ever had like a phone, I mean, not anymore, phones don't have cords. I was going to say phone cord, that ages me. But like any kind of like curly cord, maybe even if you like, if someone in your house has like a hair curler, you've seen that things, once they get wrapped around each other, sometimes they can get like kinked out of shape, and that could happen too in DNA. So it's really important that we have our topoisomerase to kind of counterbalance that effect that's going on. And all of these so far are both in eukaryotic cells and bacteria cells. So there's no real difference that we're thinking about really in all of DNA replication. They're doing the same thing. While they might have different like topoisomerase, the bacterial version, there's still like topoisomerase present. So now that we've kind of got our topoisomerase that's balancing us, we've got our helicase unzipping, our strands are separated, we're kind of moving into preparing for that actual DNA replication. So primase is going to lay down some RNA primers, and this is kind of an important thing. Anytime you're looking at a question that says, you know, it might say like predict what the primer looks like, you have to remember that it's RNA. And so you're going to be swapping in, right? U instead of U goes instead of T, or U goes instead of A, whichever one. I can't remember right now. You can tell me. Right. So you're going to making sure that you have uracil in there because it's going to be an RNA primer. Excellent. Thanks, Michaela. U instead of T. And so if you ever get asked what is the primer, they often will not prime you by saying the RNA primer, but you need to remember that it's going to be an RNA primer. So you need to look for answers that already have the U. That's a great way to eliminate other answers that potentially have a T instead. Yeah, often it will have a high GC content. We'll see that in a little bit later on. It's going to show some examples, I think. So you'll be looking for the high GC content, but you'll also be kind of looking, sometimes they'll throw it on right at the end. And so it'll be like one option or two options with a U, one of them being like if it was potentially if the DNA strand, sometimes what they'll do is they'll give you the DNA strand. So let's say this was my DNA strand, and they're asking you for the primer, right? We would be looking for something that's going to be complementary to that because we know we're making that. So it's going to be C, G, C, G. But then one of the options, right, might be TT. That's there to kind of like distract you. One of the options might be AA. One of the options might be UU, which is what we would go for. And one of them might be the exact same, but just with the UU on the end. So it might say GC, GC, UU, right? So you can see how they're going to put in options that if you're not thinking about it, you're going to go for. And that's because they kind of want, it's a way that they can ask you a question about something that is relatively straightforward, right? Knowing that it's RNA primase without exactly asking you to memorize the word RNA primase. So it's important just to remember kind of that part. I'm going to erase this so it doesn't confuse us. Then we've got DNA polymerase 3, and I would say that it's important to know the difference between the different DNA polymerases and what their jobs are, just because sometimes they'll ask you specifically about if one is knocked out or if one is not missing, or if one is like missing from the cell. How does that kind of change what's going on? So DNA polymerase 3 does most of the work. It's synthesizing the new strands off of the primers. So we know that there's going to be a leading strand, and we know that there's going to be these lagging strands in these kind of fragments. And that makes sense because, right, if this was our kind of like point of replication, we know we're always moving in a 5' to 3' direction. So we know that we're going to start here with our little primer, boop, and then we're going to kind of keep going with DNA polymerase. And when we keep going, we're unzipping in this direction. So this strand is just going to get to keep going. It doesn't really have to stop, right? The whole way down. However, when we lay another primer over here, we can really only go to that far. And we can only lay it here because hypothetically at this point, it's only been unzipped to right here. Then we're going to lay another primer right here, right? And continue in this direction, but we have to stop again. So we're having this leading strand momentum, which there's one on each side, right? This is an example of like a true DNA replication, like mitosis. If we were maybe only looking truly like for one gene that we want, we're not going to copy both sides. We'll probably just copy one side. And we've got leading again right here. And then we've got what we consider our lagging also on the other side. And they're going to be, these little sections, but they're still going to be there. And so we can see in this picture kind of like a little bit more clearly the leading and lagging strands kind of drawn out. And it is important to like recognize that the lagging strand is going to be made up of the Okazaki fragments. And so sometimes they try to like trick you with those words. They're not completely interchangeable, right? Because you could have one fragment and that's a part of the strand, and all of the fragments together equal the strand. So just like don't get too confused on kind of like those words. They don't necessarily mean like wildly different things, but sometimes you'll see them used interchangeably, and sometimes they will try to make a distinction between them. So just kind of like keep that in the back of your mind. But as we said, our leading strand is just going to be continuously extended on both kind of top and bottom in this case. And that's going to be happening, right? Remember, by DNA polymerase 3. Doing most of the actual transcription work. Then what's going to happen is that DNA polymerase 1, after we've got kind of all of this, we've got all of these little fragments, all of these little pieces going on. It's not really helpful to us that they're just pieces, right? In this example, we were trying to actually reproduce the chromosome in entirety. So we do need those gaps filled. So DNA polymerase 1, right? This is now a different DNA polymerase, has exonuclease activity. And so it is basically going to cut out the RNA and change it to DNA. That's really helpful. This is another time where they could ask you, after DNA polymerase 1 has like completed its role, tell me tell me basically what this section is. What's the code for that? And then you would need to pick, and you need to remember, oh, I'm in DNA now. So I'm not going to have any uracil. I'm only going to have my, regular 4 bases, and I'm matching here. So, and once again, that would kind of help you, Marissa. If you like remembered like Marissa did, you're going to have a lot of GC content because it's still the primer. So all of these kind of different like steps, while they seem kind of like, oh yeah, like by the wayside, if you feel really confident in them when you come to test day, it just comes naturally, and you're not having to kind of like put things together spontaneously or like guess between 2 options. So DNA polymerase 1 has come in, removed our RNA primers. So now we have, if I redrew it, we're going to have like our original DNA. We've got now DNA again. Then all of our little Okazaki fragments, but we still kind of have these like gaps in between. So we're going to have another friend. And I know this is like so kind of tedious sometimes, but we still have these gaps in between. So we're going to have another player come in, and DNA ligase is going to come in and actually fill in those gaps. And this is important. We need to like definitely have super accurate machinery that's doing this. So DNA ligase is really accurate. We also have, we talked about DNA polymerase 1 and DNA polymerase 3, but DNA polymerase 2 has the job of proofreading. So while these sections are either being transcribed or even in this kind of process where they're being kind of re-repaired, DNA polymerase 2 is actually coming through and double checking that everything is the exact right match. So it's going to be kind of like the proofreader, making sure that there are no errors. And we know this is really important because even with like single codon or single nucleotide base switches, we can have errors that potentially could halt the protein once we get to like the mRNA section when we're looking at different codons and things like that. So we know it's really important to be super accurate. At the same time, we also know that there's a chance that things are not going to be accurate, but it's kind of incredible when you think about like how many safeguards your cells have in place to make sure that this is going to happen properly. So we have DNA ligase, which has a really like good proofreading kind of like system as well that's coming in to repair those nicks, I guess is technically what they're called, but the gaps, and replace them with DNA. And then we also have DNA polymerase 2 that's going to proofread and make sure that everything is kind of ready in there. DNA polymerase 2 can proofread simultaneous to transcription as well as kind of after ligase. It's working kind of like overtime. It's going to come in and just like be checking constantly, because if we think about it, the cell, all of these kind of measures are in place because the genetic information is so important, and because it's going to be, kind of make break for this cell. So I think most of the time we think about it, I should have a picture later, I think it might be in, where like DNA polymerase 3 is building the new strand and DNA polymerase 2 is kind of following proofreading, but technically it can also proofread kind of at this like later phase once ligase has come in as well. So could be both. Great. So now that we've kind of like finished, we have DNA ligase has filled our little gaps. We've swapped out our RNA for DNA. We have our two, copies basically. We are left with this overhang. And we talked a little bit about this on Thursday, that potentially in this overhang, we are going to get shortened versions of DNA round after round, which is why it was important that our genes kind of had these regions on the ends of them called telomeres. And so ideally, we would have an enzyme come in to help us actually like rebuild that overhang and kind of lengthen the DNA to protect that gene, which we said was named telomerase. And that ideally would be present. However, we know that based on lecture from Thursday, that telomerase is not in every cell. So not in every cell. So we know that this is not kind of like super ideal. So we may not actually get that telomere extended, the 3' end. So think about it adding on like right here. So it's actually going to make the overhang more dramatic. So it would be like, if this is my 3' end, I would add on to my telomere here. My 5' end is still short down here, right? But I'm adding in that buffer zone. And then primase and DNA polymerase 3 again are going to lengthen at the 5' end after telomerase has been in. So now they are going to come in on this end here. I'm going to draw it in red and kind of match. So I had my original overhang that already kind of made my 3' end longer. I'm going to make that even longer to add in my buffer zone if I have telomerase present. And then my primase and DNA polymerase are going to come in and actually like extend the 5' end so that we've got kind of like a matching length. If we obviously don't have this DNA polymerase present, then we wouldn't have my additional like buffer zone. And so then the next time that I replicate that gene, I'm going to get a little bit shorter and a little bit shorter. And that's how we kind of talked about with like cell aging, that potentially we could actually like cut into where the gene is and that gene becomes non-functional. Yes, excellent. Great, Stephanie. So telomerase is like extending the original strand, the 3' end, to kind of build in that buffer zone for the next time it gets copied. Sorry, I do need coffee. Copied, and then primase and DNA polymerase are going to extend the copy strand that's kind of going to be underneath. It's nice in this one because it shows the difference in the colors. All right. So this is kind of the picture that I was referring to earlier, which kind of shows everything working at once. This example that Justin kind of depicted really nicely for us was very linear, but actually things are probably not happening in a linear fashion. And so you can kind of see here, we've got, hi mom, get to sleep there. You can just leave it. Sorry, you got DNA helicase kind of unwrapping in the center in the yellow. And then we've got our parental DNA helix. And then we've kind of got things going in both directions here. So you've got your leading and your template and your DNA polymerase kind of off to the top, like left corner it's got. And then you've also got DNA primase, which is working in there to kind of fix things as it's going along. And then also got our lagging strand kind of on this down right. This is like, it's kind of confusing, I think, to like identify all of the different players in this picture, but it just shows you that it's actually not happening on a linear basis. It's kind of happening in these like loops that are being pulled out, which is just kind of like interesting. And then you can see here, it's a really nice example of the binding protein, which is going to like actually help to build that space for things to kind of like actually happen. So that kind of ends our like DNA replication. Transcription is going to be similar, except in transcription, we're really only looking at a gene. So we're not going to make a copy of both sides. We're really only going to make a copy of one side. And this is where we're going to get to some differences. So in bacterial transcription, you can have multiple genes that are going to be under the control of a single promoter. And this arrangement is called an operon. So I think a lot of you, or maybe not a lot of the people that are here today, but a lot of people in class in general, were kind of confused on operons. And I think it's really important to remember that like the operon itself and that term means basically this. And then the lac operon is an example of those inducible genes, multiple genes that are under the control of one region. So we're going to go through lac operon. That's probably the most popular one, but there are other operons. So you shouldn't be surprised if you're like on your MCAT day or you're in a practice test and you hear about a different operon, because they do happen in bacteria. And so that's very common compared to eukaryotic transcription, which is just one promoter, one gene. So like if we have this promoter being activated, then that gene is going to be made. If it's being repressed, then that specific gene is not being made right or being transcribed right here. So another difference is just kind of where these things are happening. So in eukaryotes, transcription is happening in the nucleus. In bacteria, there is no nucleus, so it's obviously not happening there, but it's just going to be in the cytoplasm. And in bacterial transcripts, RNA transcripts are not modified after transcription. So basically whatever is produced by the like DNA to RNA kind of mechanism is not necessarily going to be modified with like introns and exons and kind of all the things that are going to be added into the DNA variation. So it's just important to remember that like there are going to be differences between those two. And so that's going to potentially affect kind of the outcome or what's going to like happen right in the variation of the products that are going to be made. So we'll see that kind of described a little bit further on. So this is kind of a region here where we're just seeing the promoter. We talked about Tata boxes the other day. And so it's important to remember that the promoters are going to be like really far upstream of the actual transcription start site. And this is because this whole big kind of like, machinery has to come and actually sit here. So these are the differences, again, between bacterial and eukaryotic. In bacterial, it's bacteria, they have no nucleus. They're very simple beings. There's not going to be a lot of like extra fancy things going on there. And so the nice thing is they just have RNA polymerase. Done. Don't have to worry about who's doing what's happening. They have one transcription factor called a sigma factor, one RNA polymerase. They're very simple beings compared to, of course, eukaryotes, quote unquote, more advanced. We're going to have different types of RNA polymerase for the different types of mRNA that we're eventually, different types of RNA that we're eventually making. So I would just like be familiar kind of with those. And we're going to have potentially multiple transcription factors kind of binding to make that transcription more likely. A promoter wouldn't necessarily, I know that like the word promotion, we often think of as like a benefit or an advantage, but a promoter basically is almost like, think about it more like I guess it's kind of like, think about it more like the front door. So, right, the front door of your house is either locked or unlocked. When it's locked, nothing is coming in, nothing's going to bind, like things just have to stay outside. But if it's unlocked, if that promoter is unlocked, then things can come inside. And then, there's going to be basically change that'll happen after. So the gene for you to get brown eyes or like green eyes is already determined by your parents when they've, the gamete has been made, and then whatever kind of is contained in there, whatever's dominant or recessive is going to happen. And eye color, while we think it's like just a one gene kind of thing, it's probably actually a multi-gene kind of thing. So it's going to be like all these different genes combination to then produce the pigment in our eyes. But yeah, think about the promoter more as like the front door, and whether the promoter we say like is basically like, what do we call it? If it's basically turned on or off, if it's activated or repressed, those are the words I want. So if the promoter is activated, the front door is unlocked. If the promoter is repressed, the front door is locked. So think about it more as like that way, but it is kind of like, it's almost like the signal of like, hey, this is where you need to be for this certain gene. So it is a little bit like, I guess in a way like localizing. So we know that we've got potentially, these are some of our differences that are going on, just really clearly drawn out. And so the kind of other term that we've talked about was this operator. So an operator is something that's specific in bacteria that also has activators and repressors. So whereas we talked about in eukaryotes, they do not have operons. Operons specifically basically are like control regions for multiple genes. Whereas in eukaryotes, we only have one promoter to one gene. So if you see the word operon, you should be thinking bacteria. Those are your friends. That's where you're sticking. You have multiple genes that are about to come out. If it says promoter region, you're more thinking eukaryotes, one gene is going to be made. So that's probably going to lead to most likely like one product at the end. So these are going to be, this is another option, even after my RNA polymerase has now bound in my bacteria cell, I now have this operator, which is maybe going to even prevent me from moving forward or like and completing transcription. TSS, by the way, is transcription start site. I think I labeled that on another slide. Excellent. No, that's okay, Marissa. Exactly. If we come back to our definition here of operon, multiple genes under the control of a single promoter is called an operon. So that's like part of it is that like we have to remember anytime that we see the word operon, we should be thinking multiple products, right? It's going to be multiple genes, it's multiple products, or in a bacteria, we're thinking like they've got to somehow be related. And typically they are, and we'll see that in the lac operon gene. The two products that are made like help each other out. And so typically like we're going to see on the same gene things that are related. You're not going to see like a gene for a channel protein and a gene for like some other random protein, like that don't actually have to do anything. Typically, like we're thinking about kind of like if we're gonna make these together, they're probably gonna work together, kind of thing. Does that make more sense, Marissa? Got it. Okay. So we've got our operon, potentially that's going to inhibit or allow things. And you can kind of see in this picture, remember that when we're thinking about DNA in our bacteria, our DNA is going to be this nice circle, right? So it's going to look different than potentially our chromosomes of DNA. I know these are like super simple drawings. So we're always kind of thinking about how those like are going to be setting up. So whereas in the bacteria, our bacterial example, our operon or operator, sorry, that could also be activated and repressed is going to be like right next to it. Potentially because we have a bigger complex and other proteins, right? We're thinking about the binding proteins that are going to be able to join. In our eukaryotic example, we can have kind of our activator and repressor like zones elsewhere because of the formation of like keeping those kind of strands apart and thinking about it. It doesn't have to be like, yes, it's still like a linear chromosome, but it can kind of like have its own personality because we could have kind of the other like zone basically like completely all condensed up and we're just kind of pulling apart a certain zone. Michaela, is this a way that bacteria can increase their resistance to antibiotics? Are you talking about like operons in general? If you are, I wouldn't necessarily think about like operons having like necessarily kind of maybe like I mean yeah so they can create multiple products. Yes. Okay. Yeah, it's kind of related to like any protein sequence in the cell, but particularly ones that are going to be regulated, and we'll see why in the lac operon, like why it has to be regulated or kind of how that happens. If it's if this bacteria always needs to produce something that's antibiotic resistance, it's probably not going to be under an operon. It's just going to be like allowed to be freely made all of the time. So I think about like operons, like the kind of key things to think about are like regulation, or that this is something that potentially is like needs to be made at certain times or under certain conditions. It's not necessarily going to be a protein that needs to be made all of the time. Yeah. All right. So we kind of like talked about the differences there. So when we're doing transcription, remember that we're only going to read one strand. We're not going to be looking at both like we were in DNA replication. And we've got RNA polymerase this time because we're going to be making an RNA strand, whereas before we had the DNA polymerases, which were making a DNA strand. So I'm just going to label this little friend for us. And in eukaryotes, our RNA polymerase 1, 2, or 3 is telling us what type of RNA we're making. And so that was kind of labeled on the other slide before. We're going to pretend like we are in an animal cell. So we're using RNA polymerase 2, called 2. And then this template that's coming out is not yet considered mRNA. It's just going to be considered like the RNA kind of version of going along our template strand. So you can see here when you're talking about, sometimes it'll give you the template strand and then it'll ask you for sense, right? And remember that sense is going to be really similar if I call this like my RNA transcript. The reason that this is what makes the most sense, which I know is bad to use the same word twice, but what makes like logic in my mind is the fact that this RNA transcript is going to be very similar to this strand. So that's why this is considered like the sense strand, and the positive strand is because this RNA transcript and this sense strand are going to be similar. And that makes sense because this template strand and this coding strand used to be the same, like they used to be basically complementary to each other. They are complementary to each other than they used to be. And so we're making another complementary strand to this template. So that's why this one becomes sense strand. So sometimes they will just give you the coding strand and they'll ask you for what the RNA transcript is, or they'll give you the coding strand and they'll ask you for the template, or they'll give you the template and they'll ask you for coding. So it's just like important to kind of like be familiar with these words. Sometimes then they throw in, oh, here's the template strand, give us the antisense strand. You should already think like, oh, template and antisense are the same thing. If it means when you start your bio section, you draw a quick picture of this to remember it, if you know that that's kind of a weak spot for you, then do that. But being really familiar with these words is going to help you read passages faster, and it's going to help you feel more confident when you get to a question to eliminate other answers that don't seem to make as much sense. So once we've kind of got our RNA transcript, we're going to skip over the questions just for the sake of time. There's different ways that it can terminate. I don't think these are like super important, but I included them so that you can kind of read over those and correct Marissa, because remember that we're making an RNA transcript. Excellent point. We're going to have, basically it's going to be the sense strand, except instead of thymine, we're going to have uracil. So that's another great point where they could mix you up, because they would say, here's the coding strand, what's the RNA transcript going to be? And they will put an option that is exactly the same with thymine, and then you'll be like, or yeah, thymine, threonine, whatever that base is, nitrogenous base. My brain is not working this morning. And you would know like, oh no, I don't need T's. I actually need uracils. So that's a great point to recommend. It's basically going to be terminated with different things, but what's more interesting, we're going to skip the questions, is going to be kind of how we actually get from mRNA. So we talked about here, we had our coding strand. It's considered like pre-mRNA, or sometimes called the RNA transcript, right, of what we just made, because technically we were doing transcription, so we can use that word in our template strand here. So we can see how you can see like the differences in the bases. These didn't perfectly line up, but I do promise they're like actually the same amount of letters. So you can kind of like practice with these different examples and just like really test yourself. And you'll see that if you've started taking practice tests, you know that these kind of questions are kind of snuck in. So it's just good to like be really familiar with the terms that are going to be associated with each strand, remembering that coding is also sense, and that's going to be positive, and template is antisense, and that's considered negative. Those are just, once again, like random terms. There's nothing positive about it. It doesn't have to do with the charge. They're just terms that are used. So this is something that's going to be unique to eukaryotic cells, is this kind of like, once we have the RNA transcript, this like post-transcription modification. And I think the easiest way, if you've heard about like introns and exons before, is to remember that exons get expressed. So that might help you if you feel like you sometimes like confuse which one is actually going to be, because like to me in my mind, intron sounds like it's going to be in, but it's actually not. It's going to be out. And so these exons, you're going to have a spliceosome, which is another little kind of like molecule that we need to know. And it's going to come and basically cut out our different introns. We're going to add our 5' cap over here, which you can see kind of depicted right here. And then we also add a poly A tail on the 3' end. And these are all kind of like steps in order to like process to what will actually be mRNA. So in this like form where we have the introns still in here, we're not considered mRNA. And that's another time where they can ask you a question and say like, well, what, you know, like, which is the mRNA strand? And they'll give you all these different options. And we have to remember that mRNA, we'll see later, is going to be the final finished product. Yeah, that's another good one, Michaela. So we cut out our intron here, and technically these get discarded. They all get put together and they just go to like the trash basically. And it's called a lariat, which some people like, there's a lot of research on this. It's not a super common word, but maybe if you're like struggling with vocab, that's just kind of an important one to throw in there. And it's just basically like, think about the introns being the trash. It's like a bunch of little like protein or like genetic segments of RNA. So now that we've got our kind of fully mature RNA, which I'm not really sure why it's kind of pictured so I think it's still like considered spaced apart. They haven't like truly stuck it back together in this picture, but fully mature RNA, we only have exons. We have to have the 5' cap. We have to have the 3 poly tail. And it usually basically like has these like buffer zones on the ends that are going to kind of like protect our coding region. And usually they are like regulatory, or it's where the start codon is going to be, or something like that. So that we like the ribosome will read that and know it's kind of like in the right place or doing the right thing. If there's an error already in that untranslated region, then that's going to signal for the ribosome not to make that protein. So once again, these are just kind of like checkpoints. Everything is trying to like make the right products instead of potentially making a product that could be harmful to the cell or anything like that. Another thing that is unique in the way that eukaryotic cells happen is that there could be alternate splicing. So all this means is that basically the arrangement of the exons, or the like inclusion or exclusion of certain exons, will obviously change our protein product. If we have, here you can see the colors kind of make it really clear, but 3 different exons, 1, 2, and 3, that's going to make protein X, whereas exon 1, exon 3, that's missing a section. And so it'll miss those kind of amino acids, obviously, in the sequence, and it's going to form a different protein. It's going to bind and interact a different way. Maybe there's a lot of like sulfides in these 1 and 3. And so it's going to be like a hairpin or something like that. It's going to have different kind of interactions. So this is just something to like think about in why there can be variability in kind of products that are coming from this single pre-mRNA, and how that is actually possible. Excellent. Introns are always going to go to the trash. We're not even going to like think about what's happening in there. And then dependent on the pattern or the organization of the exons, and it's always going to be linear. You're not going to see like exon 2 over here. They do kind of like tend to splice, but sometimes we'll leave certain ones out, maybe intentionally or unintentionally, but we can get kind of alternative products from our different mRNAs. Yep. This is the picture that I was kind of talking about earlier. So we've gone from our DNA that's going to be in our nucleus, right? This is also assuming that we're in a eukaryotic cell because we have a nucleus. So we've made here what we call like our RNA transcript, or potentially our pre-mRNA. I think maybe AMC uses pre-mRNA more regularly, but you'll definitely like find their pattern as you do like questions and stuff like that. We're adding our 5' and then our poly A tail, and then we're going to do our splicing. So once again, we're getting rid of those introns and basically gluing together our exons in whatever way. And then finally, we will export it out into the cytoplasm. And now we finally have mRNA and we're ready for translation. So translation kind of has like 3 phases, I would think about it, but it's not necessarily like there's a lot going on, but it's also really simple. So it's kind of one of those topics, I think, that seems like it has a lot of meat to it. But once you feel comfortable with it, it will come just really naturally. So we think about like kind of the initiation, which is going to be like the ribosome binding, getting all settled in, looking kind of for those different features like the 5' end, the GPP cap, and then also like, finding its little start sequence that it's looking for. And that's basically just like the first little like section, getting comfy, getting ready. Then we move into elongation, which is exactly what it sounds like. We're going to elongate our, peptide chain. We're putting all the amino acids on. We're building something. And then termination, which once we get to our stop codon, we're going to basically like take our mRNA out, and then our peptide, product is going to be there, depending on kind of different signals. Maybe that's happening in the cytosol. Maybe that's happening at the rough ER. We're kind of going from those 2 categories, and that's considered like termination. And our beautiful protein is sent out, and our mRNA basically gets trashed. It goes into like a ubiquitisome, or like basically like a recycling little organelle in the cell. So when we look at kind of what are going to be the factors that are going to help that initiation type connection, there is a term and a sequence called like the Shine-Dalgarno sequence. You might have heard of it before. I think it's just a term to be good to be familiar with. And that basically is how the um, small ribosomal subunit and eventually the large, because it'll come on top of it to meet, is going to know where to properly align. Obviously, it's looking for things like the GPP cap and things like that, but it needs to know kind of like we talked about that untranslated region. And the Shine-Dalgarno sequence is going to be in that untranslated region. It's kind of like a buffer before we really start doing the actual work, but it's going to help us align correctly so that we know where to start reading. So this is like points where if you potentially had a mutation in your RNA transcript, that it could be really like detrimental here. And it's relatively like recognizable ag-ag-u. So it's just like a good one to know and be familiar with, and know that basically that's like our little like, geolocator that's saying like, hello, bind here. So once we're in place, we bring our first tRNA, and this is going to be like the first step of initiation. We're then going to have our large subunit that's going to bind on top. We've got a lot of other, initiation factors, which are kind of helping getting everything lined up. Remembering that we're having, obviously, if they're hanging out there, they're going to have binding, connections and things like that. And we're in the right kind of like zone in order to get set up. And so we're going to find our start codon, AUG, put our first tRNA that matches that, and then we are going to start kind of building. So we know that methionine is always going to be our little start codon. This is another point where if you had kind of an error in your chain and it knows that like it can't really get started. It's got all these kind of checks and balances to make sure that our eventual protein product is actually what we want it to be. And this is why when we think about kind of like cancer cells or other cells that are doing things that they're not supposed to be, or like even like viruses and bacteria, when they can basically hijack our machinery to make their own product, there's a lot of steps that for regulation that like they have to be able to be smarter than. And this is why mutations happen, and why some mutations are beneficial and why some mutations are not. So we see this with COVID right now. It's trying to basically smart everything that we've done, and like exposure and natural immunity and vaccine immunity. And so these are all going to be kind of like things that if it doesn't start with a start codon, that mutation is non-beneficial. So it's not like that basically vaccine or viruses use us as their host. So if we don't replicate it, we don't get sick, we don't spread it, that mutation wasn't beneficial, whereas some other mutation has a start codon, all good to go, potentially will be. So it's kind of important to like think about these like steps similar to like glycolysis when you're thinking about like the committed steps. What are the steps in this that are kind of the regulation steps? And if they go wrong, what could happen? The MCAT like loves to like test like, basically, are you understanding the concept enough to be able to apply it in this new and different way? So we've got our start codon. It's going to be methionine, and we're ready. This is just like a zoomed in view of our tRNA and our mRNA. And just kind of like a little side note, this last position here is called the wobble position. And that's because our tRNA is like coming down to bind, and it's going to, make sure that each of the spaces in the way that they're read, and they're being read from 5' to 3'. So it's going to match A to U first, then U to A, and then it's going to match G to C. But this wobble position, we can see kind of in different regions. Like here, if the first letter is C and the second letter is U, they're all going to be the same. If the first letter is C and the second letter is C, they're all going to be proline. Sometimes it's different, right? So that's when it does need to be really specific in that third position. But here at the same, first letter C, second letter G, they're all going to be arginine. So we have wobble in that position, and it doesn't necessarily need to be like a perfect match because it's going to make the same product anyway. So the tRNAs are kind of smart. If there's like, low availability or something like that, they know that they can kind of like use that third position as an advantage. probably in biology, you saw kind of one of these codon charts before, but I would feel really comfortable using this and being able to read a sequence and predict the amino acid sequence. I've seen that on MCATs, and that just feels really good to like do that and do it really confidently and do really well. So if you haven't practiced ever with this kind of chart before, I would definitely recommend that. And you can see highlighted in this chart, it will not be highlighted in the MCAT chart most likely, but we've got our A, U, and then G, methionine, first one. And then you've got your three kind of like stop codons, which Justin gave some like really good examples of how to remember them. You are annoying, UAA, you go away, UGA, or you are gone, UAG. So it's really important when you're thinking about kind of what different kind of like codons to look for, that you're remembering that these are stop codons. And they're usually like in the chart that they give you, they're not listed as stop codons. It'll just be like black, but you won't know. So will the start codon often the time, or it'll like say methionine. So sometimes people get confused between like what is start, what is stop, where am I going? So I'd just be comfortable with these, be comfortable kind of using this chart. This picture just shows, once again, kind of that wobble position and the fact that basically once we know if we have CC as our first codons, it's always going to be proline in this example. So this third position can just kind of be like, it doesn't have to be a perfect match. If this is a C, this could be a U up on our anticodon, because it's going to produce the same thing. This tRNA is only going to be carrying a proline. It's not going to be carrying anything else. So this is kind of like just an extended example of this. So now once we've completed initiation, we've got our ribosome bound, we have, our first start codon, and we've got the methionine attached. We're in this, you can see they've got these three different sites in the large ribosomal subunit. And you are going to have to know kind of what each of the sites means. So I like to think of, and they just call them like E site, P site, and A site. But I like to think of the P as the peptide site. So that is where the like tRNA is going to come and actually like have the, or like where our peptide chain, basically you'll see once it extends a little bit is going to be kind of like growing. The A site, I like to think of as the accepting site, because that's where the new tRNA is going to join. So if I'm reading in this direction. Oh, I can't even spell. Wow. Right? I'm reading from 5' to 3'. So I read AUG, that was my start one. When I'm reading the next one, UGC, I'm going to come and kind of match it there. And so that's where my new tRNA is going to be arriving, which is my transfer RNA. It's called that because it transfers, you'll see shortly, basically the amino acid to be a part of the peptide site. We're going to see shortly that our E site is going to be our exit site, and that's where our tRNA is going to be leaving. So if we kind of go through this, it's like a nice little like stop motion kind of graphic, but we've got them here. So once we've got our second tRNA ready, it's going to bind over with our first one. So we've got our first one. So this is, think of this as like the start of my little peptide chain, and I'm going to build down in this order. We've got our next one, and then they're going to shift over so that everyone is here. We now have our peptide, right, growing in our P site. E is for exit. That tRNA is empty. It's going to leave. And we've got our next one arriving to come in. And it's got our next, we read our little codon here. We match our tRNA and we're good to go. And you can see here, like some of them are not matching perfectly. And I'm not entirely sure why they're all called UAC here, but I wouldn't like worry about that. I would just kind of like cross that out and ignore it. Typically our anticodon is going to match our codon here. So not sure what Justin was going for in this. Might just be an accident. So then the same thing basically is going to happen again. Our tRNA left our exit site. These two are going to move over to attach on to our next peptide. We can see that here. Now we have MCA, then we're all shifting to the left to kind of reshuffle. and the process continues. So it's just kind of like a nice little stop motion, you could say. And eventually we will get to a stop codon. So this tRNA that binds is going to be a stop codon. So there'll be nothing that's attached to it. And that signals for our ribosome to kind of take that chain, dislodge it, and let it be free. Does the tRNA arrive at P have first to serve as the base before the tRNA? Yeah, so think about like in the P site, that's kind of like where the first start codon comes to build the peptide. So even though it does look like it moves to the A site to kind of build the peptide, it's tricky to like depict really well. But I would just think about it like it's all kind of like shifting. Like think of them as like almost like a string of beads. They're moving together in a way, and like this product is being formed, and it's going to grow kind of in the P and A site as they're shifting and moving along. Then like as linear steps, as this kind of like stop motion little graphic shows, which I know is kind of tricky. But you can maybe watch some like other videos just to see kind of like the variability. Obviously, like all of these, kind of like graphical representations are just what we think to our best bet is going on. Some of these things, like we don't know, exactly how they're going on or like what this really looks like. We've just chosen a ribosome to like be this shape. We've chosen our little tRNAs to be this shape. It's as close as we can get, but it's probably not, 100% accurate. So think about it. Yeah, it's kind of like the P is for the peptide, because once our stop codon comes over, which let's see if there's a stop codon. So here we go. Is there one after this? Oh, excellent. So there's a stop codon here. And so we're not actually adding, like it's not going to move to the A site, Stephanie, if that makes sense, to move back to the P. At this point, this stop codon basically signals that like you need to cut this off and our little protein is ready. And Justin was very cute and did one that was MCAT as the protein. So we're technically a peptide chain. And then about like peptide folding. Yeah. So that's kind of like how that little process works and just goes along, which you can always like rerun through it and just like kind of watch the stop motion go back together. So technically, there are two possibilities for ribosomes. You have your free ribosomes, which are just going to be translating in the cytosol. And then remember that we do have bound ribosomes on the endoplasmic reticulum in the zone that we consider the rough endoplasmic reticulum. So the amino acid moves from the A site to the P site. Think about it like it will move. Yes, it's always going to be like growing in the P site. That's where our kind of like longest chain is going to be. And then once we get to our stop codon, it gets released from the P site. Does that make sense? Great. All right, so we had our free ribosomes. They're typically going to be making proteins that end up in the cytosol. Potentially, maybe will end up in the membrane, but mostly are like doing jobs like within the actual cytosol. We talked about this a little bit on Thursday, but if we have this SRP, signal recognition particle, that signals for that ribosome to actually go over to the endoplasmic reticulum, embed itself in the endoplasmic reticulum, and then that protein will actually be made to end up inside of the endoplasmic reticulum, because afterwards, this is like signaling that this protein needs to go to the ER, which is funny. It's like it needs to go to the emergency room, but it needs to go to the rough endoplasmic reticulum. It's going to be processed there, just like it would be processed elsewhere. So that we know we've still got the same tRNA kind of involved, same thing going on. Because afterwards, it's going to be potentially modified, what's considered a post-translational modification. It's going to go, we know that kind of like process of moving from the ER to the Golgi, and then it's most likely going to be excreted from the cell, or it's going to end up on the plasma membrane. So you can kind of see in this page is just like a really nice summary that free ribosomes are typically making either cytosolic proteins or nuclear proteins if they've got like a special little sequence on them, and anything that has this SRP, signal recognition particle, is going to go to be a bound ribosome, and then that's going to end up either being excreted or in the plasma membrane. So just kind of like some differences in the process, but obviously similar. They're being put together the same way. The sites are the same thing. There's no kind of like other real differences that are going on. So we're going to move on to like our little lac operon, and that's going to be the last thing that we're going to go over. I know we're kind of right at that 1 hour mark. I wrote out the steps if that's something that makes more sense for you on the following slide, but I'm just going to kind of like show a like little example here. So we know that in the lac operon, basically at rest, there is like a controlling region here, and you could just like add it on over here. So this is kind of like there is like, this is, you know, kind of like the front gate to your front door. So it's not quite, we were talking about the promoter as the front door, but now we have like a gate around our house. So that's going to be our controlling region. And there's actually going to be a little repressor that's sitting on the controlling region. So nothing can even bind. The RNA polymerase cannot even get on there to even think about producing this like stretch of genes. But however, in the lac operon specifically, when lactose enters the cell and like lactose is around, it's doing things. Maybe you just had a glass of milk. Well, this is a bacteria, but maybe this bacteria is like in the presence of some lactose, and there is going to be just like a little bit of lactose that somehow gets into the cell. When lactose, which I'm going to put in purple and just draws like L's. When lactose comes and binds, and even just like a really small amount of lactose, this means that actually there's, once we have things binding, we know there's going to be a conformational change, and that forces this controller to actually like conformationally change and move off of the gene. So it's now going to be, after the lactose is present, this is just going to go back to our like controlling region is going to be completely clear, or I can draw it down here. Right, and we don't have anything blocking the way. So what we call these two like items together, basically, that are the protein that is bound to the controlling region is called the repressor. Right, and it's just a protein. Its job is to sit there right on the controlling region and hang out. It's preventing it from being blocked. And our lactose, because it induces the conformational change, is called the inducer. And that's just like a terminology thing. If it was a different operon, we would have a different repressor and a different inducer. Those are kind of like pairs that go together. So once this repressor has moved off, it's bound to lactose, it's like hanging out somewhere else. This allows then the gene to be like to begin transcription. So RNA polymerase is going to come on, it's going to bind. Remember, we have to have like kind of all these certain things in organization, but because we know we're in a bacteria, we're in an operon, we actually have two genes that are going to be transcribed. So gene X and gene Y, and they're going to produce different products. So in the case of lac operon, and it doesn't matter like which one comes first, it's just that we know that these two products are being made. We are going to produce beta galactosidase, and then also permease. And what's important is that beta galactosidase is actually going to break down lactose into glucose and galactose, which are going to be much more usable sugars. And permease actually will go up to the plasma membrane. And it is actually like a protein that is going to insert itself into the plasma membrane to let more lactose into the cell. So now, right, we started with a really small amount of lactose in the cell. Now we have something that can actually digest the lactose, and we have something that's letting it in. So initially, when we're just letting it in, we're going to have really high levels of lactose. So that's going to keep this repressor off of the gene, right? There's tons of lactose circulating in the cell. So we know that it's bound to lactose. We're making so much more galactosidase. We're making so much more permease. Eventually, we always know that kind of things come back to a balance. And so my beta galactosidase is going to be converting my lactose into glucose and galactose. And so therefore, after a while, my lactose concentration is going to decrease, right? It's eventually going to decrease so much. There's going to be so much beta galactosidase that I don't have any more lactose. And I even need to pull off the lactose from the repressor so that I can even use that to try and get some more energy. But we know that once that repressor is free, it's going to undergo a conformational change. And where does it like to be when it's in its free state is going to be bound to the controlling region, and our transcription is going to be turned off. So that's like why kind of like operons are involved in this regulation during certain kind of environments. So it might ask you, like, in the presence of a small amount of lactose, what's going to happen? In the presence of a large amount of lactose, what is going to happen? And kind of play different scenarios. And then also, like, oftentimes we can knock out genes to see how they also affect kind of like these operon systems. If you knocked out the repressor and you didn't have any repressor, we'd basically constantly have it on. So we're always going to be making galactosidase. We're always going to be making permease. We're going to go through these kind of like troughs and waves of high lactose, and then it's all digested and high and digested and high and digested. So to probably like even out eventually. If they made permease, like basically a defunct protein, and we couldn't get any more lactose in, then potentially what's going to happen if we made the beta galactosidase, we're going to eat our initial lactose, and then we're going to kind of turn off transcription really quickly. There's a worksheet that I'll send in the chat, which kind of like helps you practice all of these different things, if I can put it, different kind of scenarios. And it's just like really helpful to kind of work through if you're feeling a little bit more confident. So I'll send that through. Oh, sorry, actually, I only sent it to Lexi. Let me send it to everyone. All right, which you might have seen this before. It's like a Berkeley review one. So Michaela said it's kind of like positive feedback. Yeah, it is kind of like positive feedback to a point, right? And then once it gets to too much, it basically turns itself off, which is like a really good kind of like control mechanism. So that is the lac operon. Very kind of like simply. There's also a really good video, which I will send out in kind of our post notes. But that is kind of the end of our lecture today. There are a few other like questions. You can just do those on your own. I'll send out answers. And yeah, any other questions that I can answer? I'm actually just going to look at I'm going to stop sharing my screen just for a second. Oh, wow, 12 people. Okay, let me just take a quick note of who's here, because I'm actually only going to send the answers to those of you that came. Oh, Jonathan, you're in here twice. Lexi. All right, any other answers that I can answer? Don't forget that I have office hours Tuesday morning, 9 to 11 Pacific time, and then we'll do the kind of glycolysis cellular respiration review 2 to 4 on Tuesday. So big review day. Any other questions? Yeah, I would just know that the sigma factor is like a transcription factor, but it's in bacteria. Yeah, of course. Hopefully it was helpful. Hopefully it was just like a nice review to really kind of like get down those foundational things. Definitely try to start the AMC practice questions or AMC practice tests, get that exposure. I will send the video via email. Yep. Have a great day, everyone. Bye. Happy studying. Make sure you get outside. Have a nice break.